So for today's lecture, we're going to have a bioenergetics review. Just a couple quick announcements. The updated syllabus is on Sakai. The only thing that was changed is that the case study number 2 is due March 20th as per last class. This is changed from March 27th. As for lecture capture, yes, it's happening. Thank you for voting on that. And each lecture will be uploaded within a week of the actual in-class lecture. So today's lecture content, we're looking at the integration of what you guys know as exercise physiologists and turning that towards an eye on skeletal muscle metabolism, and at the back end we'll have draft day. So we'll start off with a review of exercise physiology basics. First thing I want to talk about is energy balance. So energy balance is comprised of energy in as food and drink intake, as well as energy out, which is basically metabolic and cellular function, so resting metabolic rate, as well as physical activity. So we can consider either positive or negative energy balance, where positive energy balance is typically resulting in weight gain or obesity, or even the state of pregnancy. So I like to think of positive energy balance as the Christmas holidays. You guys probably went home. Your weight may have increased due to whether it's increased energy in, so more food intake, or decreased energy out, so less physical activity. On the other side of the equation, we have negative energy balance, where we can typically see this as weight loss or infection or illness such as cancer, cachexia. So how I like to think of this is you might be New Year's resolutioners, where your goal is weight loss. You can achieve this through either a decrease in energy intake, so decreased food or drink, or an increase in energy expenditure, so increasing physical activity. What actually comprises our total daily energy expenditure, so really focusing on that energy out side of the equation, we're thinking about three main factors. Resting energy expenditure, which makes up 60 to 75% of our total daily energy expenditure. Basically, this is the energy needed to keep the body alive at complete rest. So if we did nothing else in a day besides sit on the couch, our energy expenditure would be comprised of our resting energy expenditure. Next, we have the thermic effect of food, which comprises 5 to 10% of our total daily energy expenditure. This is related to all the processes involved with ingesting a meal, so that's digestion, absorption, or even transport of nutrients. And lastly, thinking about physical activity, which makes up 15 to 30% of our total daily energy expenditure. Basically, skeletal muscle contraction, we know ATP breakdown costs energy, and because of this, it results in energy expenditure. The reason why it's so variable is that we can have people whose physical activity ranges from just walking to class, whereas other people we can have on the range of elite or even varsity athletes, who would obviously be having a much greater physical activity component and greater energy expenditure related to physical activity. So what are the factors that influence resting energy expenditure? A couple things we need to consider here. So when we're talking about resting energy expenditure, we're talking about that resting metabolic rate or the basal metabolic rate. First and foremost, genetics. You might be endowed genetically with a higher or lower basal metabolic rate. Next, we're thinking about age, so young versus old. Young people have much greater skeletal muscle mass than older adults, which contributes to a greater resting energy expenditure. When we consider sex, men have a greater resting energy expenditure than women, and this can be attributed again to a greater skeletal muscle mass in men compared to women. When we consider body composition, again, we need to think about skeletal muscle mass, in which those with higher skeletal muscle mass compared to fat mass will have a greater resting energy expenditure. And thinking about exercise or fitness, again, with relation to skeletal muscle mass, we see that individuals who exercise more are more fit, have more lean mass, more skeletal muscle mass, and have a greater resting energy expenditure. So how do we tie this all together? Really, all metabolic processes generate heat, and the heat that we produce can be measured as energy expenditure. So how do we actually do this in a laboratory setting? First and foremost, you guys have probably heard of direct calorimetry, in which we measure the heat a person generates in a sealed chamber. Basically, heat energy is measured as a change in temperature, and the heat produced is proportional to the amount of energy expended. So, more heat produced, more energy expended; less heat produced, less energy expended. This is, of course, not a feasible method for most labs, as it's extremely expensive to have this metabolic chamber, and only a few in the country even have it as a research tool. More importantly, things that you guys have seen in your KINE 3P90 labs, for example, is indirect calorimetry. In this method, we're able to estimate energy expenditure by looking at oxygen consumption and carbon dioxide production. The principle is that as the body's energy expenditure increases, the amount of oxygen produced and the amount of carbon dioxide, the amount of oxygen consumed and the amount of carbon dioxide produced by the aerobic energy system increases. So, higher energy expenditure, more oxygen consumption, more CO2 produced. Lower energy expenditure, lower oxygen consumption, and lower CO2 production. This is measured through something called open circuit spirometry. Basically, we use a metabolic cart, which you guys have seen, to perform these measures. What about animal studies? So in animal studies, we have something called the Comprehensive Lab Animal Monitoring System, or CLAMS. Basically, it's a form of indirect calorimetry for animals in which they're housed in cages that can measure the amount of oxygen they're taking in, the amount of carbon dioxide they're producing, so that we can get an indirect measure of their energy expenditure. So what do we do with this information now that we have oxygen uptake and carbon dioxide production? We can use oxygen uptake to calculate energy expenditure, which is expressed as kilocalories, or kcal. So how do we go from liters of oxygen, which we can measure through a metabolic cart, to this kcal? What we need to know is that there's something called a thermal equivalent of oxygen, which is about 5 kilocalories per liter of oxygen. This is regardless of fat, carbohydrate, or protein utilization. And what you'll see is that whether it's more towards the fat side or more towards carbohydrate utilization, this number 5 will shift towards about 4.8 and towards about 5.1. So for example, if we have an hour of exercise, 60 minutes, with a VO2 of 3.8 liters per minute, we can use this calculation, so VO2 in liters per minute, multiplied by our oxygen equivalent, or our thermal equivalent of oxygen, multiplied by the amount of time, we can calculate energy expenditure. So in this case, 3.8 liters per minute multiplied by 5 kilocalories per liter, that's our oxygen equivalent, multiplied by 60 minutes, regardless of fuel used, you'll approximately expend 1,140 kilocalories. What I want to highlight in red here is that the VO2 is an important part of this equation. So what about CO2 production? So we measured this with our metabolic cart as well. We have a couple assumptions that need to be made in order to use our CO2 production for this energy expenditure calculation. First, the source of CO2 production is mitochondria, so thinking back to our TCA cycle, we know that we use oxygen in order to produce reducing equivalents through the ETC, and a byproduct of this is CO2. Second, we need to think about whether the conditions are steady state. In order for this assumption to be made, or in order for us to use this calculation, CO2 production must be at steady state. So you guys remember back to some of your exercise physiology labs. If we're exercising at a fixed intensity and we're able to produce the energy we need through aerobic metabolism, we consume a fixed amount of oxygen and produce a fixed amount of CO2 for a certain power output. So we need to meet steady state conditions in order for us to be able to calculate energy expenditure accurately. If these assumptions are not met, then we can assume that the CO2 exhaled is not reflecting substrate oxidation. Examples of scenarios where you would have changing CO2 values that are not steady state would be 1, hyperventilation, so increasing our breathing rate, we're increasing the CO2 production, or recovering from intense exercise where CO2 production would be decreasing over time. But again, how do we use that CO2 to actually calculate something meaningful for energy expenditure? That's where we turn to something called the respiratory exchange ratio, or RER. in which we calculate this as VCO2 divided by VO2, or carbon dioxide production divided by oxygen consumption. This can reflect the blend of fat and carbohydrate oxidized, but it assumes that there's no protein oxidized, and this is because our predominant fuels during exercise are carbohydrates and fat, and protein is only utilized in situations of extreme metabolic perturbations such as starvation. But in a typical, normal, healthy scenario, we are not oxidizing a large proportion of proteins. It's largely fat and carbohydrate. So here's an example in which a person consumes 1.5 liters per minute of oxygen and expires 1.2 liters per minute of carbon dioxide, and they're exercising for 30 minutes. We can go ahead now and calculate their energy expenditure. So now we know RER is CO2 over O2, in which we get 0.80. Remember, each RER has a corresponding oxygen equivalent that will dictate our percentage of carbohydrate and fat being utilized at this exercise intensity. So if we go to our equation here, or go to our chart here, what we see is that our RER is 0.8. We can go to this non-protein RQ to find 0.8. And the caloric equivalent is 4.801 kcals per liter of oxygen, or 6.001 kcals per liter CO2. To calculate energy expenditure over time, we turn back to our equation that we mentioned before, in which it's VO2 in liters per minute times oxygen equivalent in kcal per liter, multiplied by the amount of time that we're exercising in minutes. We can do this with either VO2 or VCO2, but we'll start off with VO2. So in the O2 side of things, we have 1.5 liters per minute from the last slide, multiplied by our oxygen equivalent, which is 4.801 for the RER of 0.80, multiplied by 30 minutes, we get 216 kcals of energy expenditure. On the CO2 side of things, we can calculate this by essentially multiplying 1.2 liters per minute of CO2 by 6.001 kcal per liter, which is our CO2 equivalent, multiplied by 30 minutes. Again, on both sides of this equation, you get the exact same answer of 216 kcals per energy expended, regardless of whether you used oxygen or carbon dioxide to get this answer. So 24-hour races are a real thing, so running for literally 24 hours, or fast walking, whatever you want to call it. If we wanted to calculate the total daily energy expenditure of these individuals, we can actually do that, in which we take 216 kcals per 30 minutes, make that into 432 kcals per hour, multiply that by 24 hours, and we have a total daily energy expenditure of about 10,368 kilocalories. So really, where does this table come from? You guys can see that the RER only spans from 0.7 to 1.0, so it seems kind of odd. And how is this supposed to tell us the percentage of carbohydrate or fat that we're actually utilizing? So RER varies for each macronutrient, so each macronutrient requires a different amount of oxygen consumed relative to the CO2 produced. And remember, RER is VCO2 over VO2. Basically, if we go back to our chemistry roots, think about stoichiometry, we can look at the oxidation of glucose. So for carbohydrates, we're looking at C6H12O6, which is glucose. And in order to break that down into carbon dioxide and water, we require 6 oxygens and 6 carbon dioxides. So when we go to put that into our RER equation, what we see is that we have 6 carbon dioxide divided by 6 oxygen, which is equal to 1. So 6 divided by 6 equals 1. Whereas if we take fat, we're looking at the oxidation of palmitate. So if we oxidize palmitate, so C16H32O2, requires 23 oxygen to produce 16 carbon dioxide and 16 water. So if we put this into our RER equation, 16 CO2, 23 water, 16 divided by 23 is 0.7. That's why our scale only ranges from 0.7 to 1.0, because basically the oxidation of pure fat is only equal to 0.7 and the oxidation of pure glucose is 1.0. So by that, we can use RER to tell us the relative contribution of fat versus carbohydrates towards energy expenditure. So now we're going to shift gears a little bit and talk about bioenergetics. So basically, in a nutshell, this is what we'll be covering in the next 2 weeks with regards to skeletal muscle metabolism. The goal of metabolism is essentially to store and produce energy. I want you guys to pay attention to where specific events are occurring within the cell. So first and foremost, glucose metabolism or glycogen storage breakdown, as well as glucose transport and its subsequent breakdown into pyruvate occurs in the cytosol. Interestingly, skeletal muscle contractile apparatus are also located in the cytosol. But when we consider mitochondrial or fat metabolism, we have to think about the idea of bringing those free fatty acids into the cell, into the cytosol, then transporting them across mitochondrial membrane, where the TCA cycle occurs inside the mitochondria, and the ETC is in that intermembrane space. So moving forward, I kind of want to talk about the idea of what is energy. So adenosine triphosphate, or ATP, is this high energy molecule that allows cellular processes to be carried out. It's our cellular currency of the body. And before we can talk about carbohydrate and fat metabolism, we should review basically how we use ATP to provide us with energy, or how we use it as a tool to create muscle contraction or even other cellular processes. So first, ATP can be used for chemical work. Its function is maintenance and growth, and it's really used for the formation of compounds. So anabolism is really building up, or being anabolic is to build up compounds. An example of this is muscle hypertrophy. So in response to resistance training, we have an increase in individual muscle fibers, an increase in their contractile protein content, as well as an increase in their muscle size. So basically, ATP can be used to activate the mTOR pathway, so the target of rapamycin, and this promotes muscle hypertrophy. This is something we'll review more in depth when we go into the protein metabolism lectures. And again, this results in increase in the number of muscle fibers, as well as an increase in the muscle fiber size. Alternatively, we can have mechanical work. So in this case, we're thinking about cross-bridge cycling, where ATP hydrolysis promotes the movement of that myosin head, creating that power stroke that's going to generate force for skeletal muscle contraction. Lastly, thinking about transport work. So there's a difference here between diffusion and active transport. So diffusion is basically passive and occurs down a concentration gradient, whereas what we're talking about requiring ATP is active transport, because it's occurring against its concentration gradient. An example of this would be the sarcoendoplasmic reticulum calcium ATPase, or SERCA. SERCA is involved in active transport, where following skeletal muscle contraction, calcium is taken back up into the sarcoplasmic reticulum for storage and release upon the next contraction. So this is active transport because the sarcoplasmic reticulum contains 10,000 times higher calcium than the cytoplasm itself. So basically we have to move calcium against its concentration gradient, which requires ATP as energy. So now we need to think about how ATP actually performs these steps, like how does it provide energy for these processes. So when we think about it, we need to think about the fact that ATP is made up of adenosine and 3 phosphate groups, or triphosphate. What's happening here is basically these 3 phosphate groups are linked together, but imagine sticking 2 negative charges against each other. It's like moving a magnet towards one another. There's this repulsion when the charges are the same direction. So with these negatively charged phosphates, basically they're repelling each other, and what's happening is they're creating a buildup of tension that upon ATP hydrolysis, or the removal of one of those phosphate groups, this free energy is then released. So here I want to talk about where we get ATP from metabolic pathways. Really the highlight of this slide is that glucose metabolism produces much fewer ATP than fat metabolism. And really I want to highlight the goal of the Krebs cycle in fat metabolism as well as carbohydrate metabolism is to produce reducing equivalents, which can then fuel the electron transport chain. So now thinking about the fuel stores in the human body, we should consider that carbohydrates can be stored in the muscle, that's our predominant store, the liver, second most abundant store, and smaller concentrations can be stored in the blood. For fat, we need to think about the main storage source being adipose tissue. Muscle triacylglycerides or intramuscular triglycerides make up our second largest store, and plasma-free fatty acids, we have some in the circulation there as well. So the reason why we have limited fuel stores in skeletal muscle is largely because, one, glycogen molecules stored in that muscle are fairly large and branched out. They take up a lot of space, and the skeletal muscle itself is a finite space. So now I want to talk about fuel utilization during different exercise intensities. We know that fuels are basically essential for both rest and exercise. We need ATP during all times of the day. But when we're at low exercise intensities, we're relying on plasma-free fatty acids predominantly, and as that exercise intensity increases, we're relying more heavily on muscle glycogen stores as well as plasma glucose. The reason why we rely more heavily on glycogen at higher exercise intensity is because we're having a faster rate of energy provision. So coming back to this figure here, why would glycogen be providing energy faster? Not only are the enzyme kinetics different, but also thinking about location. So glucose metabolism, again, is occurring in that cytosol right near our contractile proteins, whereas fat metabolism, we mentioned it has to enter the cytosol, then be brought into the mitochondria, where it goes through the electron transport chain and the TCA cycles, and then brought back out to our contractile properties with the ATP that we need for skeletal muscle contraction. So now thinking about fuel utilization at different durations of exercise, we have to see that at the onset of exercise, we have a greater reliance on muscle glycogen or glucose. But as that exercise intensity or the exercise duration progresses, our increased reliance on plasma-free fatty acid occurs. This is because as exercise duration progresses and steady state is achieved, we're able to produce the energy we need through aerobic metabolism and the production of ATP through fat breakdown. So a couple things I want to consider here when we're talking about muscle fiber characteristics. Just quickly thinking about our different skeletal muscle fiber types. So first and foremost, slow oxidative type 1 fibers, predominantly used for aerobic energy production because we have a high mitochondrial content. Where we consider our type 2 oxidative fibers, our type 2As, these are for oxidative phosphorylation and glycolysis, or aerobic glycolysis. These fast oxidative fibers have a lot of mitochondria, meaning that they can promote fat mobilization as well, or fat utilization. When we think about our fast glycolytic type 2B fibers, we're thinking about fewer mitochondria and predominantly producing energy through glycogen breakdown and glycolytic pathways. Another thing I want to touch on is skeletal muscle plasticity. So when we're thinking about whether the skeletal muscle is plastic, what we mean is that it can adapt and change depending on the stimuli we provide it with, whether that is exercise training or disuse resulting in atrophy. Skeletal muscle is plastic because we can have a change in the number of muscle fibers, we can have a change in the fiber size, we can have a change in mitochondrial content, the enzymes, activities that are present, or even the fuels or substrates that they use for energy provision. One thing I would like to touch on, since some of the papers that we'll be presenting are related to rodent models, we need to think about rodents versus humans and how their skeletal muscle differs. So in rodents, they have slow oxidative, fast oxidative glycolytic, and fast glycolytic fibers. They're very distinct from one another, whereas humans are more likely to have mixed muscles. So we have type 1, type 2A, and type 2B. Our range is much smaller for skeletal muscle. In that regard, when we think of a skeletal muscle in humans that we think of as a postural muscle, so gastrocnemius, this muscle is only 57% type 1 fibers, meaning that the other 43% is type 2 fibers. Whereas a muscle that we think is explosive, glycolytic, we think of biceps brachii, this muscle is really only 60% type 2 fibers, meaning the other 40 is type 1. Whereas in a rodent, we can isolate soleus, which we know is slow oxidative, or the extensor digitorum longus, which we know is a fast glycolytic muscle. So now I want to do a quick review of enzymes because the regulation of carbohydrate and fat metabolism to produce ATP requires a lot of enzymatic reactions, so we need to talk about their function, the types of reactions, and the enzyme kinetics associated. So enzymes, first and foremost, quick overview. Basically, a substrate binds to an enzyme to create an enzyme-substrate complex. Once this enzyme-substrate complex is formed, the enzyme can actually release the substrate and actually release a product as well. And again, this process can repeat over and over again, creating a number of products from that enzyme. So enzymatic reactions, we have two main types. One is a non-equilibrium reaction. So these generally proceed one way. So substrate A plus substrate B creates product AB. Pyruvate kinase is an example of this. We also have near equilibrium reactions. So these can proceed either way, so substrate A plus substrate B can form substrate AB, or can form product AB, and product AB can also be broken down into its individual substrates A and B. Basically, these near equilibrium reactions are regulated by the concentrations of substrates in products. An example of this will be creatine kinase, which we'll talk about more in the ergogenics section of this course. So now we need to talk about the regulation of non-equilibrium reactions. So these non-equilibrium reactions can have two major forms of modification. First is covalent modifications, where we can actually add a phosphate group. So phosphorylation is the event that's occurring, which can either turn on or turn off that enzyme. An example of this would be pyruvate dehydrogenase kinase, and we'll touch on that in the carbohydrate lecture. These covalent modifications are a coarse level of control because it's either off or on, whereas these allosteric modifications, in which a ligand actually binds to the enzyme to alter the speed of the enzyme's activity, either increasing or decreasing that enzyme activity. Basically, this binding of this ligand results in conformational changes, which can then influence that enzyme activity. An example of this would be pyruvate kinase. What I want to highlight is that is a level of fine-tuning. These allosteric modifications offer a fine-tuning level of control, so slight increases or slight decreases in that enzyme activity. But really, when we talk about non-equilibrium reactions, these modifications cannot reverse a non-equilibrium reaction. They can only control the rate at which they occur. So thinking about enzyme kinetics here, so allosteric regulation, again, I want to talk about the idea of speeding up or slowing down that reaction, in which we can have an allosteric activator, which speeds up that reaction, promotes the binding of that substrate to the enzyme, or we can have an allosteric inhibitor, in which binding of that allosteric inhibitor prevents the binding of the substrate or slows the binding of that substrate to the enzyme complex. Basically, allosteric regulation, what's happening is we're changing the sensitivity, not the maximum activity of that enzyme. So we can have positive and negative control. So positive effectors speed up, or that's our activators, or we can have negative or inhibitors that slow down that reaction. And basically we can have multiple allosteric effectors that can exert control on an enzyme. So more than one can be happening at once. So the last thing I want to talk about in this lecture is kind of drawing back to big picture. You guys are physiologists. I just want to consider what's limiting during endurance exercise. So the factors we have to think about are respiration, so taking that air in to our lungs and delivering it to our bloodstream. Central circulation, so thinking about once we have oxygen in our body, we need to think about how we deliver that oxygen and that blood to the peripheral tissues, so thinking about cardiac output, as well as then the peripheral circulation, thinking about how we offload that oxygen to our skeletal muscle tissues, for example. And in the case of exercise, of course, we're thinking about skeletal muscle metabolism. Are the enzymes for oxidative or aerobic metabolism limiting in any way? So these are all the things that we have to consider. And although this class focuses on skeletal muscle metabolism, it's important to consider exercise as a perturbation to the whole body, and we need to think about these other factors as well. So thinking about respiration, our arterial saturation, so when we bring oxygen into the body, it's not usually limiting, so greater than 95% in most cases. And our ability to adapt and increase our respiratory rate in response to exercise stimuli is pretty dramatic in the sense that we can have the minute volume of air that we're bringing in increased from 5 liters per minute at rest up to 60 liters per minute in response to heavy exercise, meaning either the 15 4-liter jugs of milk or even 6 cases of beer. These are equal volumes that we're talking about, 60 liters here, 60 liters per minute. So thinking about the central circulation, this is where most of our peripheral limitations occur. So thinking about cardiac output being 70 to 85% of this limitation, where stroke volume is the main limiting factor, so the volume of blood ejected per beat of the heart, since maximum heart rate is about the same in recreationally active as well as highly trained individuals, and that's about 190 to 200 beats per minute. Another example supporting this point is that blood doping, or increasing the red blood cell concentration in an individual, can highly improve exercise performance. So increasing our delivery of oxygen to those tissues is a key component of endurance exercise capacity. One thing I want to highlight with regards to the peripheral circulation is that our ability to take that oxygen off of those red blood cells is not generally limiting. And lastly, when we consider skeletal muscle metabolism, the mitochondria is usually not a limitation. So with exercise training, we can have a 2-fold increase in mitochondrial content. But this translates to a minimal improvement in maximal oxygen uptake, or our endurance exercise capacity. But there are diseases in which the mitochondria can be limiting, such as McArdle's disease, in which these people have a CPT1 deficiency, meaning they can't bring substrate across the cytosol to the mitochondria for subsequent oxidation, meaning they can't rely on aerobic metabolism, really. This makes them very sedentary and exercise intolerant, and this might be something we discuss more in the lipid metabolism side of the course. So with that, on Monday we'll talk about carbohydrate metabolism, and Thursday, study design and research methods.